Good afternoon, everyone. My name is Ellen Gurley, Manager of Corporate Communications and Investor Relations at Athersys. I'd like to welcome you all to this special webinar, Rebalancing the Immune System, the MultiStem cellular platform for treating disease and injury. Today, we will be hearing from two very talented scientists at Athersys, Dr. Willie Mays, Executive Vice President and Head of Regenerative Medicine and Neuroscience Programs, and Dr. Sarah Busch, Vice President of Regenerative Medicine and Head of Nonclinical Development. Together, they will be providing an update on our preclinical programs and sharing some details on the exciting science that has been going on at Athersys over nearly the last 20 years. At the end of their presentations, we will host a short question and answer period. To ask a question, please do so by typing in the chat. As a reminder, the focus of our webinar today is science, so we will not be able to comment on any other business or financial related questions at this time. Willie, welcome. I would like to welcome you to begin your presentation first.
We've gotten these awards from a number of different funding organizations. And these organizations aren't in it to support Athersys or to validate MAPCs. They're in it to cure diseases and advance healthcare for a number of different individuals and disease states. So again, we've been lucky, but we've also understood and learned a lot about the cells from the awards we've gotten from these organizations. And we've done this in collaboration with some of the world leading translational hospitals and universities. And so we're thankful for the collaborations we've had, the support they've given us. But again, they're not in it for us and the validation of the cells. They're in it for their own research and to learn more about the indications and the diseases they care about. So we've been doing this for 2 decades now, and I would say the success we've had, the partners we've made, the partners we continue to have with a lot of these institutions and funding organizations should allay the fears of anybody out there that wants to think that what we're peddling is snake oil, because we believe that the cells work. And I'm going to go to my grave saying that the cells work as long as someone's willing to listen to me. Very quickly, again, from a platform technology standpoint, we've learned how to grow the cells, to optimize the growth of the cells, and how to manufacture the cells in very large quantities. This is something that gives us a distinctive profile compared to other allogeneic adherent cell therapies that are out there. And we can literally make hundreds of thousands of doses of clinical grade product from a single 100 ml bone marrow donation. And we have lots of intellectual property around everything we've learned along the way in learning how to mass produce the cells. We've learned how to make a more clinically applicable formulation of the cells rather than having something that required hours in a cell therapy lab to have to thaw, reconstitute, wash, and reformulate. We now just have a thaw and administer formulation of the cells. We can thaw the cells, take them up into a syringe, place them into a bag of saline, and get them to the patient in less than an hour. We've also developed, engineered, and have a prototype for a machine that basically is like a cellular ATM that could be stored in the pharmacy at any one of our clinical centers, called the SIFU, a secure integrated freezer unit, which can store the cells, warm them up upon being instructed to do so, and spit them out the front into the hands of the pharmacist, who could then go through the procedure shown on this slide. So again, other technologies that have emerged as a function of learning how best to utilize the MultiStem cellular platform. So I'm really quickly going to go over the Acute CNS Portfolio. It's the centerpiece of where we started our knowledge and how we began to utilize our knowledge in a translational way. A lot of you will have heard some of this before. We're not going to go down a lot of rabbit holes. Very quickly, just going to touch on some of the key points. And again, we're thankful for the funding agencies. I just did want to highlight, though, we wrote all of these initial grants within the span of about 3 to 4 years. And so by the time we were working on the first grant for the first indication here. For example, hypoxic ischemic injury was the first grant we received. We were also doing work in these other indications with other collaborators that I'll mention here in a couple of slides. And that afforded us the benefit of being able to learn something in one indication, one translational animal model of acute injury, and then ask the question, geez, if the cells are working that way or doing that, how can we change maybe how we're thinking about some of these other indications, the way we're administering the cells, the number of cells we're administering, the time frame for the cells. And so there was a lot of synergy that was developed off of these first 4 core grants that really, I think, helped springboard some of our thinking and some of our discoveries relative to the cells. And one of the technologies we used was basically microarray. It's looking at the nucleic acids from the tissues in the animals that we're evaluating so that we could really compare what are the differences between sham injured animals and disease injured animals, or injury in the animal, in the tissue, for example, in the spinal cord, in the stroke lesion, in the hemisphere of the hypoxic ischemic injury. And then compare that to from cell treated to saline treated and look for differences in gene expression and gene regulation. And that really is what helped us begin to understand exactly what we believe the cells are doing and how they're modulating and interacting with not just one aspect of the immune system, but multiple different aspects of the immune system. And again, I think when you're going to tackle pathophysiologies that are so large and all of the things that go wrong when you have a stroke, when you have a severe spinal cord injury or a traumatic brain injury, you better have a therapy that works in a multimodal way, in a very profound way across lots of dimensions of the injury. And we think that's one of the reasons why administering the cells early after an acute CNS injury leads to such beneficial outcomes. We're not the first people that were thinking about the spleen, and I'm not going to talk about the spleen at all except for the next 4 slides. The first observation that the spleen was critical in stroke was from the lab of Keith Pennypacker and Allison Willing down at the University of South Florida. And they basically showed in this 2009 paper, and other people at the same time were illustrating that the spleen was an important organ in the pathophysiology of stroke. But if you look along the A row at the top of the screen, the bright lime green is dead cells. So you can see in a sham injured animal, there's no dead tissue. In the middle cerebral artery occlusion surgical model of stroke, you can see that rat has lost basically half of the neurons in this cross section. But if you splenectomized animals 2 weeks prior to doing the same exact surgery, you see a statistically significant decrease in tissue loss. We did the same experiment with Sean Savitz at the University of Texas, Houston, and published this in 2017. And what you see is on the top panel, you can see that if you look at the brain 3 days after you induce the stroke, 2 days after when you either administer or randomize the animals to receive saline or cells, that there's the same amount of tissue loss. We induced a stroke, there's going to be dead tissue. And at 3 days, doesn't matter whether you got the cells or the saline, you've lost roughly the same amount of tissue. But if you look at other animals that were taken out to 28 days, you can see, irregardless of whether the animals had spleens or did not have spleens, that there's less tissue loss in the splenectomized animals, and irregardless of whether there's spleens or there's not spleens, the MultiStem treatment was decreasing the amount of tissue loss in the brain. However, this is the critical slide that I think is lost on some people. You need to have a spleen for our cells to provide meaningful recovery benefit. If you look on the left, these are all animals that were randomized to get strokes and then either get saline or MultiStem, and you can see a statistically significant improvement in outcomes in the MultiStem treated animal. If you splenectomized animals and did the same exact experiment, randomizing animals to get either saline or MultiStem 2 weeks after having their spleens removed, there's no benefit. And this frustrates me constantly when I see people in the world of allogeneic adherent cell biology and surveying diseases where they talk about cells like ours. The MSC world talks about immunosuppression. The cells work by immunosuppression. The cells do not work by immunosuppression. They work by immunomodulation. They turn off the bad stuff faster and turn up the reparative aspects of the immune system, which we believe is catalyzed in the spleen, and we believe this figure illustrates that. I'd also like to highlight, we've spoken kind of at one-offs here and there about T regulatory cells. Sarah's going to talk about a really, we believe, important aspect of our cells when it comes to the world of cell therapies relative to T regulatory cells. But I just want to highlight across 3 different acute indications, T regulatory cells. We see an upregulation of T regulatory cells in the spleen of MultiStem treated stroke animals compared to saline. We see a statistically significant increase in T regulatory signatures in the blood compared to saline treated animals in the spinal cord injury world. And we see the upregulation of T regulatory cells compared in MultiStem treated animals compared to saline treated animals in not only the blood, but the spleen in the traumatic brain injury model. So we believe Tregs are important, and you're going to hear about Tregs a little bit more in just a second from Sarah. And then just to end real quickly with some real, we think, important data from each of these 3 other acute neurologic indications. So in spinal cord, money that was granted to us by the Ohio Third Frontier. We published a number of papers in collaboration with Dr. Jerry Silber and researchers up at Oregon Health Sciences University who replicated the work that we did with Dr. Silber. And what you can see here on the left is that if you administer an IV dose of MultiStem 24 hours after inducing a spinal cord injury, you see almost immediately an improvement in gross locomotor behavior, comparing MultiStem to saline treated animals. And it's even more profound when you're looking at fine locomotor behavior, the ability to use the forepaw in very specific ways. When we look at traumatic brain injury, again, funded by a U grant from the National Institutes of Neurologic Disease and Stroke. We've published multiple papers. I believe we're up to 6 now with Dr. Chuck Cox, our collaborator down at UT Houston. Very important piece of data here. On the left, you can see a statistically significant decrease in microglial activation. Microglia are the basically the immune surveillance cells in the central nervous system. You can see in a sham injured animal, there's not a group of animals, there's not a lot of activated microglia. If you induce a traumatic brain injury, that's CCI, you can see there's an upregulation. At a suboptimal dose of MultiStem cells, we don't see any difference. But at a therapeutically comparable level of cells, the CCI-10, you can see that there's a statistical significant decrease in microglial activation. This leads to a statistically significant improvement in memory functions, one of the major side effects in traumatic brain injured patients. And then to tie some of this together, we've done a lot of work in the hypoxic ischemic injury space with multiple collaborators, published multiple papers, received the U grant funding from, again, the National Institutes of Neurologic Disease and Stroke. And just to show you, our collaborators are Dr. Jim Carroll down at Medical College of Georgia, and with Dr. Boris Kramer and Dr. Reint Jellema at the University of Maastricht in the Netherlands. And just to show you here, again, bottom panel C, you can see these are in utero injured animals. These are animals that have not yet been born. They undergo a surgical hypoxia surgery, and then we administer the cells acutely, and then 24 hours later, and you can see in these animals, you see a statistically significant decrease in activated microglia in the subcortical white matter. We see a statistically increased level of white matter and The specific protein is something called myelin basic protein, one of the major proteins that's responsible in myelinating neurons in the brain. We see a statistically significant decrease again in the IBA1 response to this antibody in the fetal sheep. Oh, sorry, I already showed that. And then from a translational standpoint, this leads to a statistically significant decrease in not only seizure number, but actually the severity of the seizures in the brain of these fetal sheep. And that's going to correlate to improved outcomes, we believe, in hypoxic ischemic injured children who also undergo seizures. And I'll talk more about seizures at the end of the talk when it comes to epilepsy. So just to wrap it up here again, we really believe that there's a lot of applicability using these cells in a subacute time frame after acute severe neurologic injuries. We really think that the use of the cells in that time frame leads to a decrease in the negative aspects of the pro-inflammatory response and a more rapid increase in the reparative aspects of the neurologic response. So with that, I'm going to hand it over to Sarah, and she's going to talk about some of the other applications of the cells. So Sarah, whenever you're ready.
Thanks, Willie. You see my slides okay?
Yep.
Great. Hi, everyone. Again, I'm Dr. Sarah Busch, and I'm going to be going through some of the most promising preclinical programs in the non-CNS space, as well as organ transplantation and a few other acute indications. And I'll just echo Willie's sentiment that we've been really fortunate to collaborate with amazing scientists across the globe over the years to advance these programs. So really excited to get to speak to you all today. And in collaboration with Dr. Robert Miller, we have investigated the ability of MultiStem to exert benefit in multiple sclerosis. Many of you may know that MS is a disease of the central nervous system in which immune cells lead to demyelination and loss of axons, which disrupts the flow of information between the brain and the body. There are already several FDA-approved disease-modifying therapies for multiple sclerosis, for relapsing remitting specifically. However, there's still a significant unmet need in the more progressive forms of MS, and the need for those therapies to really help to resolve pre-existing CNS damage. And so, as I said, we've worked in a number of different models with Dr. Miller, including what I'm showing here, which is experimental autoimmune encephalomyelitis, mouthful, also known as EAE. And in this model, we were able to demonstrate a dose-dependent benefit of intravenous administration of MultiStem, which we showed led to reduced numbers of lesions and a reduced size of lesions in the CNS. And hopefully you're going to be seeing a video here of an EAE-induced animal that was only treated with vehicle. And you can see it's a very dire state in which the animal on the left has lost function of its hind limbs and tail. In contrast, you can see an animal on the right-hand side, which has been treated with MultiStem, and you can see there's just far superior function. The animal's able to move around the cage, use its hind limbs, rear, and in general has just a much improved overall function. So this was really exciting to us, and I wanted to show a visual of really what that looks like in an animal model. We repeated that study a number of times, modifying various aspects of the experimental design. And one important observation that we made was that the timing of MultiStem administration was really important. And if we gave MultiStem cells too soon, the cells were not effective. And I think that mirrors something of what Willie showed and touched on really briefly earlier in spinal cord injury, and in which we've seen that the cells are not responding to, they're not responding to the injury, they're responding to the body's response to the injury. So we think that that's a really critical conserved aspect of how the cells are working and what they're able to sense and how they can respond. And we've also looked in a more chronic, subchronic, chronic timeframe a few weeks after the initiation of EAE. And we saw impact in this time frame, which is really encouraging, and showed us, that there was an ability to intervene at a later stage. And when we were looking more into this model, we wanted to understand how much of that was due to immune modulation and how much of that was due to more driving of repair processes. And we did full gene expression arrays across many different organs. All I'm showing you here is data from the spinal cord. And here we showed that there were a number of things called upstream regulators. That's genes that are known to drive the expression and influence other genes, essentially. And these upstream regulators were strongly influenced by MultiStem. And I'm showing you just a couple examples here to pro-inflammatory gene families and to CNS health and function gene families. So those inflammatory modulators were downregulated by MultiStem, as we would hope to see, and those indicators of CNS health and function were upregulated by MultiStem. We also examined the impact of MultiStem in a model called LPC, or lysolecithin. And this model results in a rapid loss of myelin and oligodendrocytes. Those are the cells that produce myelin in really a focal area. So just in one specific spot, so you can look really closely at what's going on in the lesion environment. And interestingly, what we saw in this model was that intravenous administration of MultiStem did not have an impact on remyelination. I haven't shown that data here. But with direct administration into the site of the injury, we saw a striking increase in the amount of myelin. And this is, really encouraging to us. But in this model, we were unable to dissect an impact of the immune response, as there's still some inflammation ongoing in the spinal cord at this time, and an impact on remyelination. So for that assessment, we moved into something called the Cooper zone model, which is more representative of the chronic setting that I was referring to earlier, and very little inflammatory component at this stage of the disease. And here, again, we saw no impact of intravenously delivered MultiStem, but we saw an increase in remyelination with the direct administration of MultiStem into the ventricles in the brain. So a slightly different approach, but it showed that the cells themselves, MultiStem, was able to drive remyelination in the absence of, that direct immune response having an impact on them locally. And finally, we researched the impact of MultiStem on those oligodendrocytes, or the progenitor cells that actually generate the myelin, provide the support for axons. And what we found was that MultiStem secreted factors actually increased these cells in vitro, and they promoted their maturation. You can see how much different they looked. They're promoting their ability to produce myelin, which we think is really exciting as a basis for further investigation, not only in multiple sclerosis, but in other diseases as well. I now want to touch just briefly on two other CNS disorders that we have done some initial work in, the first being Parkinson's disease. And again, many of you may know that Parkinson's is a progressive disease that occurs when specific populations of nerve cells that produce dopamine are injured or die. And this causes movement problems that are associated with Parkinson's disease. We used a model here in which an agent called 6-hydroxydopamine was injected to destroy this specific population of dopaminergic neurons. And the animals then were treated IV with MultiStem or vehicle, and we saw new formation of new neurons in the striatum of just the MultiStem treated animals, which was really exciting. And when we looked at the gene expression in multiple tissues, we found indications of immunomodulation that was consistent with what we had seen previously in many other disease indications that we've gone through with you today, that are really conserved across those key different aspects of the MultiStem cell platform. We've also done some initial pilot work in Alzheimer's disease, which is the most common cause of dementia. And this study was done with a grant from the National Center for Regenerative Medicine here in Cleveland. And here we utilized a mouse model called AICD, transgenic mouse. And these mice have features including tau pathology, aberrant neuronal activity, memory deficits, and neurodegeneration that show up in an age-dependent manner. So, more relevant to the Alzheimer's disease state than some other models. And here we examined the effects of, again, intravenously administered MultiStem to these animals. And we found that they had a significant impact on the activation of microglia, which is a mechanism that we've extensively examined in traumatic brain injury, as Willie showed you briefly in some of his slides earlier. But again, something that we've investigated previously, an effect on cells in the brain despite administration of the cells intravenously. So that's really interesting to us. So overall, in the chronic and progressive CNS space, we've shown that MultiStem exhibits a lot of disease-modifying impacts that we think represent opportunity for future development. So we're really excited to see what the future will bring with those indications. Now I'm going to shift gears slightly into the realm of organ transplantation and tolerance. So many of you know from our previous disclosures about our advanced clinical work in acute respiratory distress syndrome, or ARDS. Excuse me. But we've also investigated the ability of MultiStem to preserve and improve the quality of transplanted lungs and other organs, which I'll talk about in a moment. And a common cause of failed lung transplantation is inflammatory and immune mediators that can really damage the lung in the acute time frame post-transplantation. So once there's actually been a transplant. In studies, in studies with our collaborator, might have a sound here. In our studies with our collaborator, we were able to show that human lungs that were isolated from organ donors that had inflammation prior to use in prior to the lungs being used in transplantation were able to be modulated by MultiStem. And so we looked at administration of MultiStem, administration of MultiStem to one lobe of the lung and vehicle to administration of the other lobe of the lung, which provides a nice internal control in the system. And what we found was a reduction in inflammation in the MultiStem treated lungs and a particular effect on the numbers of macrophages, which are an important cell type that we've seen modulated in many different disease indications. And this is just showing you the modulation of those macrophages, kind of a summary of the reduced secretion of pro-inflammatory cytokines that we've seen throughout many disease and injury areas, and increases in reparative type macrophages, as well as a reduction in the ability of macrophages and other leukocytes to actually get into the site of injury. So we're reducing their ability to pass through endothelial cells and exert, an impact immediately at the site of injury. Also in the organ transplant space, we've collaborated with other leading researchers in the renal area at Newcastle University. And these researchers have been, focused on the impact of kidney health following transplantation. And you can see here that there's a huge number of Americans that are on the waiting list for a kidney transplant, a significant number of people dying on the waiting list for a kidney transplant. So it's a very serious unmet need. And the group in Newcastle has been working with a technique to better preserve those donated kidneys, keeping them alive outside of the body in a normothermic machine perfusion circuit. And so we were able to harness this technology and then add MultiStem to it, essentially. So we're adding the cells into the end of the circuit and then looking at the impact on the health of the kidneys. And so looking at, again, pairs of kidneys, we were able to see that the kidneys that were treated with MultiStem became more transplantable. So they're making more urine, they had better blood flow throughout the kidney, and overall just less inflammatory ischemic damage. And so therefore, we're dramatically increasing the ability to, transplant an organ and not having to worry about treating the whole patient or giving added immunosuppression. So that's something that we and the clinicians that we're collaborating with continue to be really hopeful about. This is another model of transplantation that isn't talked about too much, and this is a model of heterotopic heart transplantation. This was a really exciting collaboration in which we were able to show that MultiStem can be used to promote tolerance and acceptance of a donor heart. And you can see that what we're calling the primary transplant on the left. And, I think probably most excitingly, however, despite, the primary transplant effects were exciting, but potentially most exciting was the ability to show that when there was a secondary transplant, so taking the heart that was accepted in the first animal and transplanting it to a second animal, another naive animal, that heart was accepted with no immunosuppression, no immunosuppressive drugs whatsoever. So this was really exciting to us, and again, to our collaborators, to be able to show that we could potentially reduce the number of those, quite serious drugs that need to be given after a transplant. And in this study, we also showed the critical nature of macrophages to the induction of that tolerance of the organ, as well as T regulatory cell induction, which was a conserved mechanism. And this brings me to an important program that we've developed in collaboration with researchers at King's College London, a T regulatory expansion platform. And T regulatory cells, Willie touched on earlier, I've touched on a couple times, but this just shows you an overview of the many ways that they can impact the immune system and drive a more positive or regenerative immune response. And these cells are being developed in their own right as a novel therapeutic approach. And we've previously published a lot of work. Alice Valentin-Torres here at Athersys drove the work around understanding the induction of T regulatory cells by MultiStem. And I won't go through this in any detail today, but suffice to say, concurrent with this development of our understanding of mechanism, we developed a MultiStem and regulatory T cell co-culture platform. And again, this was in collaboration as well as with researchers at our wholly owned subsidiary, Regenerative. And so this expansion platform system, it's a bit complicated, so I won't go through it in too much detail again. However, suffice to say, current clinical trials utilizing these T regulatory cells are a challenge because it's difficult to manufacture enough T regulatory cells to give a sufficient dose. So therefore, we have been looking to drive the expansion of T regulatory cells for adoptive cellular therapy with MultiStem. And we successfully showed that these cells, which we call multi-reg, are more able to increase, they're able to increase the yield of these T regulatory cells in manufacturing and reduce the time to establish a target dose of cells that would be given to a patient. And really interestingly as well, multi-reg cells were able to be expanded from patients with autoimmune diseases compared to matched regulatory T cell lines. So this allows for the expansion of a stable and effective regulatory T cell product from many patients that are in dire need of this cell therapy. And finally, I'll give a brief overview of the work that we've done in the acute non-CNS space. We very recently announced the results of a radiation countermeasure study that was conducted in collaboration with the Armed Forces Radiobiology Research Institute, or AFRI. And we've actually been conducting research relevant to acute radiation syndrome over several years. So even back before these collaborations with AFRI and NIAID, we were working in the space of hematopoietic stem cell transplant and graft versus host disease. And we even completed a clinical study which showed favorable tolerability and promising results as well. And for those of you who are unaware, acute radiation syndrome is an acute condition that comes from irradiation of the body by a high dose of penetrating radiation over a very short period of time. And at lower doses, you see you see bone marrow damage that can ultimately be lethal without intervention. And so here we looked at the efficacy of intravenously delivered MultiStem after acute radiation syndrome and found that MultiStem cells increased the survival of animals that were treated with MultiStem, resulted in higher body weights, which I'm not showing here in those surviving animals, and positive trends in recovery of the hematopoietic system. And this histology that I'm showing is from a prior study that we conducted in the same model. And you can see the sternum of these animals treated with MultiStem demonstrating a really dramatic improvement in cellularity. So we are really encouraged by these results and plan to consider doing future studies in this area. And Willie has spoken extensively in the past in previous presentations about our phase 2 clinical study in trauma, which is actively enrolling at UT Health. This trial is comparing mortality and the incidence, severity, and duration of inflammatory complications and kidney injury, which is, I believe, a very important endpoint that we have a lot of confidence in, given the data that I've shown you across that space today as well. And now I'm going to turn it back over to Willie to go through one more disease and injury area that we're pursuing. So thank you.
Here, make sure I have the right, let's see. And there we go, full screen. So the last aspect that I'm going to talk about, I'll talk about two other platforms that we have that basically deal with non-human MAPC, as well as a novel target identification and validation aspect of the cells. The reason we learned about this, this novel application of the cells was through a collaboration we did with a large biopharma. Epilepsy is a horrible disease. There's acute forms of it. When you're having a seizure, it's a full-on neuroinflammatory crisis, but the people that have the periodic seizures live with it their whole lives. And so some people are refractory to some of the drugs. Some of the new drugs that are out there cause significant problems. When we were working with the company we were collaborating with, they used a multi-drug sort of model for inducing seizure activity in the brain of rodents. So you give a drug, you wait 30 minutes, you give a second drug, and then 2 hours later, the animals start to have seizures. You moderate some of the seizure activity with another drug. And then we waited 4 hours and 24 hours later and gave 2 doses of the MultiStem cells intravenously. And so basically there were 2 identical experiments done 3 months apart. You can see that the administration of MultiStem in red, in experiment 1 and in experiment 2, leads to a decrease in the severity of the seizures and an offset in the timing when the seizures first occurred. And when you add or you do the composite of all the animals in the bottom here, you'll get this sort of 1 week experiments 1 + 2, and you can see again the offset. And this was statistically significant. There's a number of different stages for seizure activity. The higher the number, the worse the seizure activity is in the animal. And you can see there's a statistically significant decrease in the worst phenotype in the MultiStem treated animals compared to saline, and a statistically significant upregulation in the mildest form of the epileptic seizure activity. And we also preserved, there was a lot of significant decrease in lethality in the MultiStem treated versus PBS animals. And that was great. We hit a lot of the endpoints that we were looking for in the collaboration. But after we were done with the experiment, we asked them, can you harvest tissues so we can go back, run home to mother again with the microarray experiments. And basically what we did was we got all the microarray data from the tissues we were interested in, and we had some of our lab mates in Regenerative, specifically Annalise Bogarts. We didn't tell her what was going on. She didn't know anything about the outcomes, and we just had her evaluate the microarray data from the brains of the animals. And what came back was we saw lots of changes in the MultiStem treated versus the saline treated animals. We were able to identify lots of targets that were in the literature that were well known to be targets of epilepsy drug discovery efforts across multiple different biopharmas. But that also gave us the ability, and here's an example of that. Here, these are all genes, and I have those blocked off for our own well-keeping at this particular moment in time. But you saw that our cells were affecting a lot of well-known targets in the world of epilepsy. But what the other thing was that was interesting is, as we started to go down the rank order of the genes that were meaningfully changed by an administration of MultiStem, we ended up finding all kinds of other genes that are logically implicated in epilepsy or could be novel target candidates. And then just to convince ourselves that this was something interesting, we went back and we had the same sort of analysis done on some of the MS tissues that Sarah spoke about. And sure enough, we were able to see that MultiStem was affecting a number of known targets that were being sort of focused on by some of the big pharmas in the world when it came to MS. And likewise, when we looked at some of the genes that weren't known to be druggable targets, there were a number of other genes that came up that certainly appear to be implicated in MS pathophysiology. So that's another application of the cells as a platform that we can do traditional novel drug target identification with in the appropriate animal models. And then finally, just to highlight roughly 10 to 12 years of work that were done by our colleagues over at our European office in Regenerative. This work that was led by Annalise Bogarts and David Cray. If you can find a MAPC in a human, you can likely find a MAPC and mass produce a MAPC in other species as well. And the group over there looked and found and produced MAPCs from both feline, canine, and equine donors, began to do basic disease modifying research in a number of different indications. I'm only going to focus on some of the dog work here. And this is for anybody that's had a dog and a dog that lived to be 12 or 13, you know, they start moving more slowly, going up and down stairs in a more labored way because they start to suffer from significant joint disease. This was a surgical model of joint disease. And you can see on the left-hand side that the IV administration, the intra-joint administration of MAPC compared to PBS led to a decrease in the severity scores in the histologic index that was used to grade the severity of the lesions, and that there were only animals that received canine MAPC responded in this model. And when we looked at some of the cytokines and some of the pro-inflammatory mediators, you could see that they went up as a function of time in the joints of the animals that received saline as opposed to the animals that received a MultiStem administration. And of course, we saw that there was an induction of T regulatory cells in the canine treated, the canines treated with MAPC compared to the saline treatment. So with that, we want to save time for questions. I just want to take a moment. Been a lot of turmoil at the company that I think we're headed in the right direction now, but we had a lot of restructuring and we had to let go of a lot of good people. And I want to make sure we say thank you to all the scientists here at Athersys and Regenerative for all of their hard work for 20 years. I want to thank the academic collaborators. I want to thank the funding agencies who made some of these studies possible. And obviously on the other end, I want to thank the clinical teams who continue to work with us to treat patients with MultiStem, and obviously those patients and their families. So with that, if there are any questions, I will stop sharing and see if anybody has written us anything. And I don't.
I have not seen any Q&A come
Yeah.
through yet. Ellen, do we know if it's enabled?
Yes.
there's any questions?
The Q&A portion should be enabled, yes.
Thank you. Yes. A non-scientific question is the first one, and so we won't be answering that here. Thanks, though. Yes, I got a question from a well-known, I believe, former investor in Athersys, and we did seek CIRM funding, and we sought CIRM funding twice. Once for funding in the world of the ischemic stroke trial early on, and once in the translation of the traumatic brain injury work into a clinical trial. And we received a score that was 1 point below the fundable range both times. We were in the, I believe, in the 95th percentile for scoring on both of those funding calls, but we didn't receive funding from CIRM at that time. That doesn't mean we can't go back to CIRM, and it's something we've talked about internally, but at this moment in time, we have not applied for any more funding. So a question from Greg, how will you prioritize these programs? I think that's really, considering where we are right now, Greg, a function of who knocks on the door or who picks up the phone. I mean, in the past, we were, I believe, a little more reluctant to want to partner these out on kind of a one-by-one basis, but I don't believe that's the way Dan's thinking about things anymore. So I think those are any and all of the things we talked about today, I believe, could be partnered, just a function of who's interested. Are there any trials expected to be initiated in the near future for any of these? We would love to. We would love to fund a lot of these in the clinical setting. As a matter of fact, I know the National Institutes of Neurologic Disease are fairly well chafed with me because we received the U grants in both TBI and hypoxic ischemic injury and pretty much got the ball across the goal line in both of those, but we haven't run a clinical trial because we don't have, at the time, we didn't have the pockets deep enough to run another clinical trial than the ones we were conducting. But we would certainly like to get back to those. In both of those cases, we had formal pre-IND discussions with the FDA as kind of the final checkbox to complete the grants, and we did both of those. So let's see. We do not have any grants currently submitted since we reorganized and went more streamlined. Most of our efforts really are in the world of completing clinical enrollment in the MASTERS-2 trial and having ongoing discussions with those interested in having discussions with us. Which programs would you consider most de-risked? Well, TBI is a, when you put trauma in its totality into a bucket, it's the third leading cause of mortality in the United States, and it's the first leading cause of mortality up to the age of 45, and then again after the age of 75. So that is a pretty big market, and TBI makes up a significant portion of trauma, quote unquote, in general. And it's been de-risked, and we've had the FDA, we've had a formal pre-IND, so we're ready and rolling when it comes to TBI. I have a question here that says, can you comment on dosage between disease so toxicity results? Yeah, so we've done for every one of the indications that we move forward into clinical trials, Steve, we do a full-on workup to make sure that we're not leading to anything toxic. And we've had walk-in dose escalations in most of the indications early on, just to make sure that, it's one thing to treat an animal in, for example, ARDS with your cells, but when you have a lung disease, you want to make sure that when you're giving an IV administration of something that passes through the lung, that you're not exacerbating anything.
I might add to that, Willie, too, just in terms of between the different disease conditions, that some of the diseases and injuries we've talked about today require far fewer cells, like the solid organ transplant, for example. Ex vivo treatment of just a kidney requires, far fewer cells. We're looking at on the order of 50 million cells. So it's, just a drop in the bucket compared with what we were looking at for an IV administration to the whole human being. So it definitely varies, and we're taking it into account and examining the different profiles for the different indications.
Yep. Great job. And then Sarah, there's one here from Carl that I know we talked about and thought about, but I'm not sure that there's an answer. If you want to go ahead and.
Oh, it's jumping around.
Yeah.
Sorry, 1 second, Carl. Plausible way to use IV administration of MultiStem to induce myelination for indications such as AD or PD. Yes, sorry, that's what you're talking about, Willie. We have talked about that. We've begun exploring it in a research and development fashion. There were a number of publications that came out suggesting that T regulatory cells could actually drive remyelination. Bless you, Alan. And we were very interested in that hypothesis, and it would make perfect sense with a lot of the work that we showed you today. So there's definitely a plausible way to envision that.
Yeah. Let's see. Yeah, so here, multiple doses may be optimal for treating chronic indications, ? And I totally agree, Nick. I mean, that's one of the things. Once we figured out kind of how the acute scenario was working, we ran around and chased the acute scenarios while we continued to do basic research with our collaborators and in our own labs on chronic indications. And that absolutely may be the case, that multiple dosings, and there's some other companies out there that have developed sort of devices that may make treating chronic injuries or chronic conditions that are loaded with cells but aren't implanted into the body. That may be a possibility as well. Let's see. Let's see, it's hard to read and come up. Looking at a repair and plasticity. In this respect, the brain is actively undergoing changes over a longer period of time. Can you go into detail describing the changes happening in the brain that is leading to improvements in the clinical trial stroke patients? Yeah, that's one of our hypotheses, and one of the things you heard us mention several times today, microglia. Sarah did a lot of work early before she ever joined Athersys, looking at the glial scar. Basically, there's a wall put up when you induce an injury in either spinal cord or in stroke. Axons are sensitive to the presence of activated microglia / macrophages. And when you put down something called a proteoglycan, the axons will pull away or recede. And then this glial scar is built up and basically walls off the injury from the non-injured brain. If you put we believe when you administer our cells early, you minimize or negate the formation of the glial scar so that the axons retract, but then as a function of time will eventually find their way home and be able to form a healthy neural circuit again. Let's see. What indications are you seeing the most interested in? Well, different. We've had interest in most of the things we shared today with you at one time or another. Dan asked us to put this presentation on so we could re-energize and freshen up, I think, the perspective of maybe those interested parties who never knew or didn't know all the things that our cells could do. So trauma should move quickly. We're enrolling in the second cohort in the trauma trial, and after we're done with the second cohort, there'll be a review between us and some stuff we'll have to submit to the FDA to move then quickly, hopefully into cohort 3, and then it would be move as fast as we possibly can. And I mentioned the trauma trial. That's pretty much it, I think. And we're just past the top of the hour. So I'm done, and certainly appreciative of the 100 of you that were on the line at one time. I really appreciate everybody making time for this. Sarah, Ellen, any last comments?
No, I think you covered it. Thank you so much, everybody, for giving us the chance to speak with you.
Thank you.
Thank you, everybody.